Hello, my name is Sarisha Lujan, and I am a staff global training and development scientist here at Illumina. Today, I will be going over our Illumina 5-base solution. On the agenda today, we'll start off with a product overview. Then we'll go into the assay workflow. So for the 5-base solution, we have two different kits, our whole genome and our enrichment kits, and I'll be going over the workflow for both. Then we'll move into sequencing and run planning, followed by a high-level overview of the DRAGON secondary analysis. So starting off with the product overview. So the 5-base solution is flexible with sample types and methods. For sample inputs, both kits are compatible with gDNA or cell-free DNA. For gDNA, it's a 50 to 100 nanogram input. This does require mechanical shearing prior to starting the workflow. And for cell-free DNA, it's a 1 to 20 nanogram input from plasma or serum, and shearing is not required. One thing to note is that FFPE is not formally supported as an input type. As I mentioned, we have two kits in this solution, the Illumina 5-base DNA prep, which is our whole genome kit, and then the Illumina 5-base DNA prep with enrichment, which is our targeted kit. Analysis can be done germline or somatic using our DRAGON applications. So DRAGON germline, DRAGON somatic, either in tumor only or tumor normal mode. And then for our enrichment kit, you can use DRAGON enrichment as the application. One thing to note is that we do have a DRAGON methylation application. However, it is not compatible with the 5-base solution. So this is a high-level overview of the entire workflow for both kits. So starting off with library prep, we start off with end repair and A-tailing, move into ligation, and then we get into the specific that is for the 5-base kit, which is that enzymatic conversion of the methylated C's to T's. So we first do a denaturation step, then do the conversion step, move into PCR, and then if you are running the enrichment kit, there are the additional enrichment steps. Otherwise, you move into sequencing where we pool, quant, and normalize our samples, then load it onto our sequencer, and then kick off that secondary analysis within the DRAGON applications. We do have a specific 5-base pipeline that is run that has the methylation-aware algorithms, goes through mapping and alignment. There is an optional UMI consensus, and the output is both that dual variant and methylation calling. The output of the DRAGON analysis can be input into any tertiary analysis, including our Illumina connected multiomics. For sequencer compatibility, the Illumina 5-base DNA prep is compatible with both NovaSeq systems, so both the NovaSeq 6000 and the NovaSeq X series. And the enrichment kit is compatible with both the NovaSeq systems and the NextSeq 2000. As I mentioned, we have developed a novel 5-base pipeline to be able to provide the high-quality dual detection of methylation and variant calling with exceptional speed. So these 5-base pipelines are built for the germline, somatic, and enrichment analysis pipelines. It is kicked off within those DRAGON applications. And again, it is not compatible with our DRAGON methylation application. So now moving into the reagents used in the workflow and then the required equipment. For both kits, the whole genome and enrichment, they come with these two boxes. So the Illumina 5-base DNA prep box 1 and box 2. Box 1 goes in the freezer, box 2 is at room temperature. And two reagents that I want to highlight are the GDMC, which is the genomic DNA methylation control, and the SFMC, the short fragmentation methylation control. So the GDMC is only used for the gDNA workflow, and the SFMC is only used for the cfDNA workflow. So when you're going through the workflow, depending on your input type, there will be a tube left over since you won't be using it. A little bit more details about the methylation controls. Both of them are a combination of PUC19, which is a fully methylated genome, and lambda, which is a fully unmethylated genome. So for the SFMC, or the short fragment methylation control, as I mentioned, it's only used in the cfDNA workflow. It is pre-sheared to match the cfDNA fragment size, and it's combined with the sample after the size selection step. So it's not a separate well in your plate, it's combined with every single sample. Similarly, the GDMC, or the genomic DNA methylation control, again, it's only used in the gDNA workflow. It's a high molecular weight control DNA. It's combined with each sample prior to the sonication step. So both of these are kind of put into the workflow at different steps, and of course, it's for the different input types. So addition of these controls will provide the QC metrics for conversion efficiency. If you are using the enrichment kit, you will have these two additional bags. Bag 1 goes in the freezer, and bag 2 is stored in the fridge. Outside of these two bags, you are required to purchase a custom enrichment panel, and then there's also an optional methylation control panel. So the panel that's used for the enrichment kit is an Illumina Custom Enrichment Panel V2. It is designed specifically for the Illumina 5-base DNA prep with enrichment through Illumina Design Studio. So some specifications for this panel are that it has to be 120 base pair probe length, double-stranded DNA probes, and greater than or equal to 75 kb in panel size. So if you are creating a panel that is 75 kb to 1 MB, the PCR cycle recommendations are provided in the product documentation. However, if you want to design a panel that's greater than 1 MB, optimization will be required and may require further reduction of PCR cycle number. One thing to note is that third-party panels are not supported. So for some background on the 5-base panel design strategy, the design does account for both methylated and unmethylated sites on both strands. So we can kind of take the example that we have here up in this box with the sequence. Here we're showing that it has methylated Cs on both the plus and minus strands, goes through our 5-base solution, and those methylated Cs get converted to Ts. However, there could be some methylated Cs or just on the plus strand, or the methylated Cs only on the minus strand, or no methylated Cs in whatever our input sample is. So we have both of those examples of how that would look like and how it's targeted by the probe design illustrated here. So when it goes through our 5-base conversion, if it does have methylated Cs, then there will be converted to Ts, and we do have the probe designed to target if there are those Ts that have been converted for the methylated Cs. And down here with the unmethylated fragments, we also have those probes to target the unmethylated plus and minus strands. An additional panel for the enrichment workflow is this optional methylation control panel. Again, it's available to order through Design Studio as an Illumina panel, and it's used in the targeted enrichment workflow in combination with SFMC or GDMC to provide those methylation conversion quality control metrics. One thing to note is that the methylation control panel must be diluted prior to adding it to the workflow to avoid over-enrichment of the small genome controls and kind of taking over that data quality. And I'll go through what that calculation is and at what step you're supposed to do that. And these are just some of the user-supplied consumables and equipment. You'll see on the left that for the gDNA workflow, for that mechanical shearing, we recommend a Covaris LE220 Plus or other sonication instrument that can do the sonication based on the specifications we provide. And then for the Covaris, it's the 8 microtube strip or alternative DNA fragmentation instrument consumables. On the right, you see that for the cfDNA workflow, we do have an optional quant that would require the high sensitivity qubit kit, which is not used in the gDNA workflow. The full list of consumables and equipment are listed in our product documentation under the user supplied consumables and equipment section. Okay, so now we're moving into the assay workflow. So this is a high-level overview of the whole genome workflow using the Illumina 5-base DNA prep kit. And this is the high-level protocol overview of the enrichment workflow. You'll see that it's almost exactly the same as a whole genome workflow with those additional steps for enrichment. So starting off with library prep and conversion. As I mentioned, the Illumina 5-base DNA protocols, both the whole genome and the enrichment, enable library preparation from 50 to 100 nanograms of gDNA. It's crucial to stick to this input amount because the enzymatic conversion has been optimized for this range of gDNA. So use a sonication method that produces an average fragment size of about 450 base pairs, and the sonication reaction must be less than 100 microliters to be compatible with this workflow. So Illumina has tested the Covaris LE220 Plus with the 8 microtube strip for sonication with the following settings shown here on the slide. This is also listed in the product documentation. If you are doing the gDNA workflow, we start with shearing the gDNA. So as I mentioned, you combine the gDNA with the gDMC prior to starting sonication for each sample, and the total volume must be less than 100 microliters. Sonicate the samples to 450 base pairs, and then transfer the samples to a plate. Add RSB if needed to increase that sample volume to 100 microliters. Best practices here is to optimize sonication for the sample type, sonication equipment, and sonication consumables. And a reminder that overfragmenting can reduce sample yield, introduce GC bias, and affect assay performance. So this is a safe stopping point. If you do want to stop at this point, you can store the samples in the freezer at -25 to -50 degrees C for up to 30 days. Once we've sheared the gDNA, then we go into size selection. We start off with removing the large DNA fragments by adding vortexed IPB to each sample, shake, incubate, and pellet on a magnetic stand. You retain the supernatant, transfer the samples to a new plate, and then do another cleanup with IPB beads to remove the small DNA fragments. Followed by that, we do 2 washes with fresh 80% ethanol, and then add RSB to elute off the beads and transfer 45 microliters from each well to a new plate. Best practices here is to make sure that IPB is stored correctly. You do not want to freeze the beads. Vortex IPB thoroughly before use and confirm there's no pellet at the bottom of the tube. The bead solution is viscous, so you want to aspirate and dispense slowly. As I mentioned, prepare fresh 80% ethanol for those washes and use fine tips to remove residual ethanol. Again, this is another safe stopping point. You can store the samples at this point in the freezer for up to 30 days. So if you are doing the cfDNA input instead, these are the input recommendations. So for the blood collection tubes, use one of the Illumina tested blood collection tubes, which is listed in the product documentation, just to ensure compatibility with the workflow. For extraction, use a nucleic acid isolation method that produces high recovery yields, minimizes sample consumption, and preserves sample integrity. Again, those compatible kits are listed in the product documentation. For quantification, it is recommended to quantify input using a size-based quantification method, such as the fragment analyzer or the tape station. And you want to quantify the cfDNA fraction only, which is 75 base pairs to 250 base pairs. And as I mentioned, both protocols enable library preparation from 1 to 20 nanograms of cfDNA. So if you are starting off with cfDNA, the first step is to prepare size selection of cfDNA. So it is that 1 to 20 nanograms input, similar to what I said for gDNA. The cell-free DNA workflow has been optimized for this input range to maximize conversion. So you want to stick to this range and not go outside of it. Add RSB for a final volume of 80 microliters if needed. Then we move into some bead cleanups. First, we will remove the large DNA fragments by adding vortexed IPB to each sample. Shake, incubate, and pellet on a magnetic stand. Again, we retain the supernatant here and transfer the samples to a new plate. Then we move into purification step with the IPB beads. After that, we do two washes actually, with that fresh 80% ethanol on a magnetic stand, and then we elute by adding RSB, transfer 42.5 microliters from each well to a new plate, and then here is where we add the SFMC control to each sample. Best practices here, again, is to follow the bead handling best practices for IPB that we just went over in the gDNA portion. Again, prepare the fresh 80% ethanol, and then when removing the residual ethanol, use fine tips to get it all. This is another safe stopping point if you want to stop here. There's also an option to save this cfDNA elution plate for cfDNA quantification that is at the next step. So you can either store it on ice and move into the quantification immediately, or freeze the plate and come back to the next quantification step. So as I mentioned, there is an optional quantification step for the cfDNA workflow. This is using the high sensitivity qubit kit. So you prepare that kit per the manufacturer's instructions. Place the cfDNA elution plate on a bar magnet, and then use 2 microliters from each sample well to do the quantification. So this step really can only be performed if the input is greater than 5 nanograms, just because of the level of detection that the high sensitivity kit can produce. Typical yields for a 10 nanogram input is in the 0.2 nanogram per microliter range. So now we move into the library prep workflow. So this is the same regardless of whether or not you have gDNA or cfDNA input. So the first step is end repair. And we add ERP6 to each well of the library prep plate. Seal, shake, and centrifuge the plate, and then place it on the thermocycler to run the ERP program, and then immediately proceed to the next step. So best practices here is to ensure that the thermocycler is pre-programmed with the ERP program listed in the product documentation, and this is not a safe stopping point, so you need to immediately proceed to the next step when the program reaches the 4-degree C hold. Next, we move into A-tailing, where we add ATL4 to each well of the library prep plate. Again, seal, shake, and centrifuge the plate, and then run the ATL program. Best practices here are similar, where you want to ensure that the thermocycler is pre-programmed with the ATL program listed in the product documentation. And again, it's not a safe stopping point, so you need to immediately proceed to the next step. Now we move into ligation of the adapters. We add LIGX and UMI3 to each well of the plate. Seal, shake, and centrifuge, and then run the ligation program on the thermocycler. Again, ensure that the thermocycler is pre-programmed with the LIG program listed in the product documentation, and we need to proceed to the next step since this is not a safe stopping point. So now we move into one of the critical steps of this workflow, which is the cleanup after the ligation. So we start off with binding the IPB beads to each sample. Again, shake, incubate, and pellet on the magnetic stand. We remove and discard the supernatant, then move into the 2 washes with 80% ethanol. Then there is the final air dry after the second ethanol wash. The plate needs to be air dried for 15 minutes. So this is to ensure that there is no ethanol carryover for the conversion steps. So it is critical to make sure that you do this 15-minute air dry prior to eluting the samples with MRR and then transferring that to a new plate. So again, that 15-minute dry cannot be skipped. Ethanol carryover can negatively affect assay performance. And if beads are observed in the pipette tips when aspirating, solely dispense back to the bottom of the originating well. Bead carryover can also negatively affect conversion. So, as I mentioned, this is one of the most crucial steps of the workflow to ensure that conversion step happens successfully. Both bead and ethanol carryover from the step will affect the efficiency of the conversion enzyme that we use in the next step. And methylation conversion will be lower if there is that carryover, resulting in reduced sensitivity. So after the step, this is a safe stopping point. You can store your plate in the freezer for up to 30 days. So now we move into the conversion and amplification portion of the workflow. We start off with denaturation. So we add DDR, which is the DNA denaturation buffer, to each well of the conversion plate. Seal, shake, and centrifuge the plate and run the denaturation program. While that program is running, we will prepare the conversion master mix according to what's listed on the table on the right, and you want to add the volumes in the order listed. Invert the tube 5 times to mix. You do not want to vortex this master mix, so invert or pipette mix is fine. And then centrifuge and store on ice until use. Again, this is not a safe stopping point, so you want to immediately proceed to the next step when the thermocycle program reaches 4 degrees C hold. Best practices here is to ensure precise volumes are added for the conversion master mix. Proper ratios of MCB and IME control the conversion reaction. Accurate preparation of this mix at the correct ratios balances the conversion reaction. So too little IME relative to MCB may result in lower methylation conversion, which is reduced sensitivity, and then too much IME relative to MCB may result in elevated false positive methylation conversion with the controls. Rinse pipette tip when adding IME to the master mix to ensure the full amount is added, again, to ensure that that ratio is correct. And it is crucial not to vortex IME or the conversion master mix. IME is just sensitive to vortexing and can result in reduced conversion. So then we move into the actual conversion step. We pipette mix the conversion master mix before use, and then add it to each well. Seal, shake, and centrifuge the plate, and then run the conversion program. Best practices here is to avoid introducing bubbles to the conversion master mix when pipette mixing. Ensure the thermocycler is pre-programmed with that conversion program listed in the product documentation. And again, this is not a safe stopping point, so you want to immediately proceed to the next step. So now we move into index PCR. With the conversion plate on ice, add the following in order. So the Illumina DNA RNA UD indexes and the MAM, which is a methylation amplification mix. Seal, shake, and centrifuge the plate. And then depending on which kit you are using, that's what you will be running the PCR program. So for the whole genome kit, it is the PCR6 program, which is listed in that product documentation. And then for the enrichment kit, it is the PCR10 program. Similar best practices here: ensure the thermocycler is pre-programmed, depending on which kit you're running. And then once the program reaches 4-degree hold, immediately proceed to the next step, since this is not a safe stopping point. So now we move into a cleanup step using the IPV beads. Pretty standard cleanup step here. We first bind with the IPV beads, remove and discard the supernatant, and then do the ethanol washing it twice, and then we elute the samples with RSB. Best practices here is to follow IPV best practices that we've gone over a few times already in this training. And this is a safe stopping point, so the plate can be stored at minus 25 degrees C to 15 degrees C, basically in the freezer for up to 30 days. So here's where the two workflows kind of diverge. So if you are doing the whole genome kit, you'll move into quantifying the libraries. So here we quantify 2 microliters of the libraries using the qubit broad range assay kit. And there's also an optional step to assess the fragment size using either TapeStation or a bioanalyzer. So for the quant, the target yield, if you have a 50 nanogram gDNA input, is 4 to 20 nanograms per microliters. And then for 10 nanograms of cfDNA input, it's 10 to 30 nanograms per microliter. If you do the fragment size analysis, then the fragment size distribution for gDNA is about 600 to 700 base pairs, and for cfDNA, it's about 310 to 350 base pairs. So we'll go over the enrichment workflow if you do want to use the enrichment kit. So prior to starting the workflow itself, if you are using the methylation control panel, it must be diluted before starting hybridized probe section, because you have to use the diluted methylation control panel within that hyb mix. So first, you have to calculate the dilution factor based on the custom panel size and the sample type using the equation here, where it's the dilution factor equals to 50 plus custom panel size in kb divided by 550 times the dilution constant. And the dilution constant is based on sample type, and you can see on the table on the right, based on the application type, the cfDNA is 0.01 as the dilution constant, and for gDNA, it's 0.05. So then you dilute the methylation control panel in RSB according to the calculated dilution factor. So I have an example here. If we have a cfDNA application with a 100 kb custom panel, we put those numbers into our dilution factor, and we get a dilution factor of 27. So the final dilution is 1 microliter of MCP and 26 microliters of RSB. Just a couple reminders. The methylation control panel is optional, so if you are not using it, there is a different reagent you can use. I believe it's RSB in place of that when you are making the master mix. The MCP enables monitoring of the methylation conversion using the spike and control genomes included in the kit. So that's the GDMC or SFMC. And then, again, it must be diluted to avoid over-enrichment of those small genome controls and to not affect the data quality. So the first step is to hybridize probes. For 4-plex pools, we transfer 2 microliters of each of the 4 samples into a single well of the hyb plate, and then repeat for additional samples as needed. Pipette mix each enrichment pool 5 times, and then we combine the reagents listed on the right here to prepare the hyb master mix. So you see we use NHV2, the Illumina custom enrichment panel V2, so that's the required panel that we have designed in Illumina Design Studio specifically for the Illumina 5-base DNA prep with enrichment. Then we use the diluted methylation control panel, which is optional. So if you don't want to use that, then you use RSB and EHB2. So all of these reagents make up the hyb master mix. Vortex to mix that, and then add the hyb master mix to each well and pipette mix 10 times. You'll run the Hive program and immediately proceed to the next step. So best practices here, NHB2 precipitates and separates during storage, so ensure that solution is clear prior to creating the master mix. The Hive master mix can get cloudy over time after EHB2. Repeat vortex if the Hive master mix appears phase separated prior to adding it to the samples. And again, the Hive master mix must include the methylation control panel if you want to get those methylation conversion quality control metrics. Then we move into capture. Here we bind the samples to SMB4 and then run the INK58 program. When the program completes, place the plate on a magnetic stand and discard the supernatant. Then we move into washes, 2 washes with EEW with shaking and heated incubation. Remove the supernatant after the second wash and then elute using EEW. Resuspend and transfer libraries to a new plate. You'll incubate with the INK58 program for 5 minutes, and then we proceed immediately to the next step. Best practices, make sure to use SMB4 and not IPB for this part of the workflow. Make sure that SMB4 are at room temperature before use. If SMB4 is not at room temperature, salt crystals can form. And it's crucial to transfer the eluted DNA to a new plate after the wash. This step minimizes carryover of residual reagents that can inhibit downstream PCR. Then we move into amplification step. So you prepare the PCR elution mix, which is listed in the product documentation. Vortex, place it on ice. We'll remove the plate from the thermocycler, place it on a magnetic stand, and remove and discard the supernatant. Add the PCR elution mix to each well, seal and shake the plate, and then run the EL PCR program and immediately proceed to the next step. Best practices here is to ensure the thermocycler is pre-programmed with the EL PCR program listed in the product documentation. So PCR cycles do vary depending on the panel size range, and that's listed here in the table on the right. And one thing to note is that the beads are still in the PCR plate during amplification, so that's not something that you need to remove prior to starting that program. Then we move into a cleanup. First, we do a probe cleanup, where we transfer samples to the probe cleanup plate and add SMB4. Again, we run the INK58 program, and then we bind using IPB. So during the incubation, vortex IPB and add it to each well of the PCR cleanup plate. Transfer the samples from the probe cleanup plate to the PCR cleanup plate and then incubate. And then we wash twice with 80% ethanol and elute the samples with RSB. So follow the bead handling best practices for both IPB and SMB4. And just a reminder that both bead types are used during this step, so ensure that the correct bead is used at the correct step. And this is a safe stopping point. You can store the plate in the freezer for up to 30 days. Now we're at the end of the enrichment workflow, where we quantify the enriched libraries using the Qubit Broad Range Assay Kit. And again, there's an optional step here to assess the average fragment size using either tape station or bioanalyzer DNA. So the quant target yield here is 10 to 80 nanograms per microliter. It is panel dependent, but that's the range we're sort of expecting. And then fragment size distribution, similar gDNA, is 600 to 700 base pairs. And for cfDNA, it's 310 to 350 base pairs. So that's the end of the assay workflow. We'll now move into sequencing and run setup. So as I mentioned, we have several sequencers that are compatible for the 5-base kits. For the NovaSeq 6000, which is both for the whole genome and the enrichment kits, we can use the S4 flow cell, either the standard or XP workflow. For the NovaSeq X series, the 1.5B, 10B, and 25B flow cells are compatible with the kits. And then the NextSeq 2K, which is only recommended for the enrichment kit, it is the P3 XLEAP SBS onboard workflow. So it has to be the XLEAP SBS kits and the P3 flow cell that are used on the NextSeq 2000. For all the sequencers, the run length is 151, 10, 10, 151, and 1% 5x spike-in is recommended. These are the starting concentrations and final loading concentration recommendations that are available for the whole genome kit in our product documentation. You can see it's listed for both the NovaSeq 6000, depending on which workflow you are using, and then each of the NovaSeq X series flow cells. So here are the sequencing coverage recommendations for different applications, specifically for the whole genome kit. You can see what the use case that we've tested, what the recommendation for the sequencing coverage is, as well as which DRAGON secondary analysis pipeline. One thing to note is this sequencing coverage, you can find it in the WGS metrics file, and the metric name specifically is average coverage on human autosomes. So once you've completed your analysis, if you want to see if you've achieved what we have recommended, you will find it within that metric file using that metric name. If you are doing the enrichment kit, this is the table that lists the starting concentration and the final concentrations based on your sequencing system and the flow cell type. We don't have the plexly listed here because it is panel and application dependent, so you will have to contact support to help you find that calculation and walk you through what the max plexity would be based on your panel. So once we've diluted our sample to the starting concentration, then we will do a run setup depending on how we want to do analysis. So if you are doing DRAGON on-prem analysis, there's a separate run setup workflow versus the cloud analysis. And again, the 5-base pipeline is launched within the DRAGON, germline, somatic, or enrichment applications, and it's not compatible with the DRAGON methylation application. So if you are using an on-prem server, you will first stream sequencing run data to your local storage over your local network, mount or copy the run data to the DRAGON server for analysis, then perform analysis on the DRAGON server, so running BCL convert first, then manually launching the DRAGON application plus enabling that DRAGON 5-base pipeline, and then finally copy the analysis output to your local storage. If you're doing cloud analysis, so either in BaseSpace or ICA, you can create a sample sheet or plan a run on BaseSpace Sequence Hub. So create the sample sheet manually, or use BaseSpace Run Planning tool to plan a run, or create and export a sample sheet. You'll start your sequencing run and stream the data to BaseSpace or ICA. BCL Convert, which is a FASTQ generation, will automatically launch after that sequencing run is. Then you'll have to go in and manually launch your DRAGON application of choice, plus enabling that 5-base pipeline. So one thing I want to highlight, regardless of how you are setting up your run, is the UMI trimming. So UMI trimming is required because it's critical for accurate methylation calling. Omitting these override cycles will lead to a higher than normal conversion rate for the lambda control genome, which is the unmethylated, since the indexes are also not methylated. So you can see here the example for the BaseSpace run planning. This should auto-populate However, if you are not using our cloud run planning tool, we do have sample sheet templates available on the product website, which does have the override cycle settings available and already inputted under that BCL convert settings for you to be able to use and make sure that it's there. So I will walk you through run planning within BaseSpace. You'll first start off with going to the Runs tab in BaseSpace Sequence Hub after you have logged in. Select New Run up here, and then select Run Planning, which will bring you to this page. And this is where you will enter your run details. So first we'll provide a run name. Then select your sequencer, so either the NovaSeq X series, the 6000, and if you're running the enrichment kit, you have the option to run it on the 1K, 2K. Select BaseSpace or ICA as a secondary analysis location. And if you are planning a NovaSeq X series run, this is where you enter in the read lengths, which again is 151, 10, 10, 151. Once you hit next, then you come to this configuration page. Here is where you select the application, which is Dragon BCL Convert, whatever the latest version is. Then you'll select your library prep kit, which again is either the 5-base DNA prep or the 5-base DNA prep with enrichment. And then you'll select the index adapter kit that you used. And within the dropdown are all the compatible index adapter kits that you can use with this workflow. Once you hit next, then you'll come to this page. If you are running the NovaSeq 6000, this is where the read lengths and override cycles are auto-populated. It's also where the override cycles are listed for the NovaSeq X. And one thing to note is to not remove the override cycles. Trimming is critical for analysis for this workflow, and changing these values can result in reduced methylation calling for accuracy. So leave everything as the defaults as they are auto-populated here. And then you'll move into entering your sample information. So there's a few options. You can enter in the sample information within the user interface. You can download the template that's available here, update it, and then import it, or you can import a V2 sample sheet to upload the sample information. So this is just an example of a sample template with 6 samples added to it. And then this is how the table looks once the samples are entered. Then we come to the analysis settings, and all of these are left blank, so the adapters will be trimmed, and the barcode mismatch defaults to 1. So at the end, this is what the review looks like. It has all the information that we just inputted, and then you can either export the sample sheet, or for the NovaSeq X and the NextSeq 1K, 2K, you can also see this button that says Save as Planned, which will automatically go to the instrument. So depending on your instrument, there are different ways to start the sequencing run. Whichever platform you're using, make sure you follow the product documentation that's listed here. And a reminder, after sequencing and run upload to BaseSpace or ICA is complete, BCL Convert will automatically launch. And then after BCL Convert completes, then you'll have to go in and manually launch the Dragon application, either in BaseSpace or ICA. Okay, so now launching that secondary analysis after BCL Convert is completed. As I said a few times, there are several different analysis options. We have the Dragon germline specifically for all these applications. It's the 4.4.6 version. So within Dragon germline 4.4.6, we can do human CNV variant calling, human small variant calling, human no variant calling. So this is methylation only. And then non-human no variant calling, again, methylation only, and the only supported species for this right now is mouse. For Dragon somatic, again, it's a 4.4.6 app version, we can do tumor-only mode or tumor normal. And then for the enrichment kit, using the Dragon enrichment application, we can do somatic high, medium, and low coverage, and then germline enrichment as well. So depending on which application you'll be using and what type of variant calling you're doing, specific settings will need to be enabled. The walkthrough for each application and variant calling type is available in the Dragon documentation online. The most important step, which is what I'll be going through, is to enable the Dragon 5-base DNA pipeline within these applications, because otherwise the analysis will just not include any of the methylation calling. So if you're launching the analysis in Base Sequence Hub, this is for all applications. One of the tabs that is available now is the methylation for Illumina 5-base DNA prep. So you'll expand that, that tab, and then you will select this checkbox that says Enable 5-base methylation aware algorithms. Again, you have the option of launching analysis directly in ICA. There is this section called Methylation Options for Illumina 5-base DNA Prep, and you will select True for Enable 5-base Methylation Aware Algorithms, and it kind of explains what the algorithm is doing and what Dragon options are enabled. As I mentioned, the 5-base is a part of Dragon Core, and so you will be able to run the Dragon 5-base pipeline on your on-prem server. To enable these 5-base command lines, though, you must be upgraded to the Dragon 4.4.6 version. And then these command lines that are written right here need to be included to enable the Dragon 5-base pipeline. So again, depending on which application you're running and what variant calling you want, there's a whole list of all the different command lines that need to be run and enabled, and you have to make sure that these 3 lines are also included to enable that 5-base pipeline. And then finally, there are some input files that need to be added as well, depending on which application you're running, and if it's on the on-prem server. So for the Dragon enrichment, you will need the target bed file. This is what defines your panel's custom target regions. And then if you're running Dragon somatic or Dragon enrichment, the somatic variant calling within that, you'll need a baseline systematic noise bed file. And then finally, if you're running the analysis on your Dragon on-prem server, regardless of application, you will need the FastQC list CSV file. Okay, so now moving into a high-level overview of the 5-base pipeline specifically. This is an overview of the Dragon 5-base DNA pipeline that integrates genetic and epigenetic analysis. Again, this pipeline is what enables simultaneous genome and methylome profiling through specialized processing options. So it's a pretty typical workflow for our Dragon analysis, where we start with the FASTQ files, using a hash table for map and align, standard positioning, sorting, and duplicate marking. If you are running the enrichment kit, this is where you put in the optional target bed file, and where we also have our optional UMI collapsing if that's something you want to select. And then here's where we split out to getting both methylation calling and then the methyl-aware variant calling, depending on what variant calling you have selected. And the output files are the GVCF, VC variant calling metrics, and again, the methylation metrics, as well as the M-bias file. So the pipeline summary, we have a 5-base hash table. So a 5-base reference hash table is stored under a subdirectory, which Dragon Mapper uses automatically. The map and align of the 5-base data follows the standard Dragon DNA approach, which is in the Dragon documentation. The Dragon 5 base algorithms have been updated to account for the possibility of a C to T conversion due to methylation at both seed mapping and alignment scoring stages. The methylation calling for methylation is identified by a C to T or a G to A mismatch, with variant calling deconvoluting methylation from true variants. And specifically for the small variant calling, this pipeline supports both germline and somatic variant calling on the 5 base data with enhanced algorithms to distinguish methylation-induced changes from variants. Finally, there's also the option for CNV calling, supported for whole genome sequencing in germline and somatic contexts with some limitations, which is listed in the Dragon documentation. So a little bit more about how the pipeline distinguishes methylation from variants. It does leverage information encoded in the complementary DNA strand to distinguish between the two. So using the reference that is listed here at the top in this box, we can differentiate between a true variant, where it's a C to T, versus the methylated locus. As we go through the workflow, with the true variant, you can see that the final alignment to the reference, it has T both in the plus and the minus strand. And one thing to note is that the last minus strand sequence here on both the variant and the methylated locus is the complement of the input minus strand aligned to the reference, just to really highlight the differentiation using that complementary DNA strand. And then on the right, you can see here the methylated locus that has a methylated C on both of the strands. Once it goes through our workflow, and then it's aligned to the reference, there is one strand that has the T variation versus the A variation. And this is because of the unique nature of methylation, where there is always a C before a G, and we can tell apart those C to T mutations from the methylated loci. The C to T variants, the variant pattern is present on both strands, as I showed here. And for the methylated locus, the variant of C to T is only found on one strand, while G to A is found on the opposite strand. So now we'll discuss the secondary analysis outputs and some key metrics. So if you are using the base user interface to look at the results for your secondary analysis, you can navigate to the Reports tab, and you will find at the top a consolidation of all of the samples into one report, or you can look at it at a per sample level if you want. And you'll see there are various tabs here across the report. And the last one is specifically for methylation. It contains alignment metrics, fragment metrics, and then also some of those QC conversion metrics that I talked about earlier in the assay workflow, which we'll cover in a little bit on where to find them and what it looks like. Under the Inputs tab, it has all of the information of what has been selected to kick off analysis. And one thing you want to make sure is that this Enable 5-base methylation-aware algorithm has been selected. So the final tab that's available in the base user interface is the Files tab, and this is the output folder structure that is available in that Files tab. So at the top, we start with the output directory, whatever that's been named, and then there are folders for each of the samples containing that sample ID from your sample sheet. And then within that, there are all of the files that are available for that sample. There are a lot more than what I have here. These are just some of the key output files that I wanted to point out. So starting off with the CX report, this is the genome-wide cytosine methylation report that contains the methylation status of every reference cytosine in the genome. Then we have some of our common files like the mapping and alignment metrics, the coverage metrics, the methylation calling metrics, which does contain the methylation specific metrics, the M-bias metrics, And then one thing I wanted to highlight is the GVCF or VCF, depending on which analysis you have kicked off. Methylation reporting can be integrated into the VCF and GVCF output files, and methylation reporting within these files is provided for the reference allele and the alternative alleles. And I'll go into all of these files just a little bit more in the subsequent slides. One thing I wanted to highlight too is that these are the key output files that are inputs for tertiary analysis in Illumina connected multiomics. Going into all the files a little bit more, so the cytosine report is, as I mentioned, a genome-wide cytosine methylation report. It contains the methylation status of every reference cytosine in the genome with non-zero coverage. So the position and strand of each C in the genome are given in the first three fields of the report. The C context in the reference is given in the sixth field, and then the number of reads are in these two fields, depending on whether or not it's a methylated C or a regular C. And then finally, the trinucleotide sequence context is given in the last field. As I mentioned, the methylation reporting is integrated into the GVCF or the VCF, whichever analysis you are using. So information from the CX report is also available in those files with additional insights. These formats provide a compressed single file output for dual-omic results. The methylation-specific CX report is still included for compatibility with legacy pipelines, and we demonstrate high concordance between the CX report and the GVCF or the VCF. The GVCF or VCF is recommended for high accuracy genome-wide CpG methylation reporting. So there are several metric files. The mapping metrics, which contains mapping specific metrics, the coverage metrics, which contain key metrics about read coverage over a specified region, and finally the methylation calling metrics, which contains methylation-specific metrics that are generated for methylation calling, including benchmarks like total number of cytosines analyzed, count and rate of methylation in each cytosine context, strand of the best alignment, etc. Two metrics that I wanted to highlight within the methylation calling metrics file are the methylation control QC metrics. So this is when GDMC or SFMC is added to the workflow so that we can get these methylation control QC metrics. So on the left, you can see how to navigate to it through the BaseSpace UI. Under that methylation tab, you'll scroll down to the context QC metrics. And here you see multiple samples within the same report. If you want to look at it at just per sample level, then there are separate files for that. But you can see here the lambda unmethylated and the PUC19 methylated controls are listed, and we have the percent of CpG listed right here. So that's how you navigate through the BaseSpace UI. If you are looking at the individual files themselves, you'll navigate to that methylation calling metric file, which ends in .methyl_metrics.csv. And under the methyl QC column, you can see the lambda unmethylated control, and then a little bit lower is the PUC19 methylated control, and it's specifically the percent of Cs methylated in the CpG context, which calls out the same values that you can see here in the BaseSpace UI. There are some other metrics to look at for library quality or if it's something of interest for you, or something that we've referenced in our literature as like the metric name, and it doesn't necessarily match up with the metric name in the file. So what I've listed here is the metric name, what the description is, and that output location that you can find them, and then what the metric name is in the file, just in case it is abbreviated or something like that. So this one is specific to the whole genome kit, and we have metrics that are listed both in the WGS metrics and then the ones that I just covered, which are in the methylmetrics.csv. And on this slide, we have the common metrics for the enrichment kit. And again, it's the metric name, description, the output location, and then that column name. And a little bit about Illumina Connected Multiomics. So there are some resources that are already available. There is some demo data and a walkthrough that is listed in the Illumina 5 base documentation that's linked out here. And then there are other resources online specifically for ICM, and there's even a demo of ICM on YouTube. Additional on-site training is available for purchase. You should contact your sales representative for more details. But for the Illumina 5-based DNA prep, it is a 2-day on-site training where we go over the workflow and then walk through with hands-on training with the kit and load it on the sequencer. And then for Illumina 5-based DNA prep with enrichment, it's a 2.5-day on-site training. And that concludes our training. Thank you for joining.